Day 2 of the 27th ASGCT annual meeting continues here in Baltimore. I'm Linnea Olivares, incoming chair of the ASGCT Communications Committee, and I'm honored to bring to you this next soundbite from Dr. Chuck Murray, fresh off his presidential symposium talk called Improving Human Cardiomyocyte Therapy with Genome Editing and Metabolic Reprogramming. Dr. Murray is co-founder and director of the Institute for Stem Cell and Regenerative Medicine at the University of Washington School of Medicine, but he recently announced that he'll soon be transitioning to be the new head of USC Stem Cell at the Keck School of Medicine, chairing the Department of Stem Cell Biology and Regenerative Medicine, and directing the Eli and Edith Broad Center for Regenerative Medicine and Stem Cell Research. Wow. Congratulations, Dr. Murray, and thank you for joining us today.
Thanks for having me.
So I actually wanted to take a step back in time for a moment. When you first envisioned this big picture concept of heart regeneration many years ago, what did you have in mind?
Well, I had in mind actually that we could reprogram the injured cells, the fibroblasts in an infarcted heart, and make them change their identity. This was totally biting off more than we could chew, because it turned out that we hadn't even sequenced the genome. We didn't know all the genes involved in cardiac development. So we figured out how to turn an injured heart into skeletal muscle, which was interesting, and it said that cells are potentially plastic. But it took another 2 decades before we had sufficient knowledge of the genome to know what we needed to do to really reprogram.
And you've propelled this field forward from both industry and academia. But after transitioning into industry as CSO at Sana for several years, you decided to go back to fully focusing on the academia side. And you don't see that every day. What informed this decision, and what more do you hope to accomplish in your new position at USC?
Sure. And just to be clear, I wasn't chief scientific officer. I was the head of what was called the cardiometabolic cell therapy. So I oversaw the heart regeneration and the diabetes program. I don't want to take someone else's role. But what propelled the change back to academia was really the change in the financial markets. That the company I worked with, Sana Biotechnology, was not able to continue development in both CAR T cells and many of these other more challenging regenerative therapies. And so they ended up spinning my program back out. And thank goodness for tenure, because I had a job to go back to at the University of Washington.
What are the most significant changes you've observed across the CGT ecosystem over the past decade?
Well, the biggest thing has to be that we have clinical data now, finally. And if I may, the ASGCT really kept the flame alive during a couple of tough decades. When any society can get together when there's great clinical data and lots to celebrate and things like that, but when there's a vision that hasn't yet been realized and it's a long journey, it's really hard. And the ASGCT is what really kept things going. I think kept the enthusiasm in the field and kept people coming together with the sense of optimism that we can do it.
And you talked a little bit this morning at the conclusion of your talk about the challenges still remaining to realize that full vision. So bringing it full circle to where we started this conversation a few moments ago, what's your vision now for the future of organ regeneration and that impact in human health, and what do we have to do to really realize that?
I think the future is bright. I see a time where these scourges of humankind, heart failure, yes, but kidney failure and chronic obstructive lung disease and retinal blindness and deafness and all kinds of diseases for which we have no good therapies at the moment, they all have cellular deficiency as their root cause. I see the time where we're going to be able to replace critical populations of most cells in the body, and this is going to greatly extend human health span.
That's so cool.
It's cool, right? I mean, it's like science is fun, and it's a great way to make your living. And I mean, every day I wake up and I get paid to be curious.
Oh, I love that. You need to frame that somewhere. So just a quick plug. I saw this morning you're hiring in your new lab. Do you want to talk a little bit more about what you're looking to build out in your team?
Yeah, so the University of Southern California has some really bold visions that they think now is the time to take serious shots on goal for both gene therapy and for stem cell-based therapeutics. And so I'll be overseeing their efforts to bring stem cells into the clinic. And of course, the line between gene and cell therapy is a blurry one, as it should be, because why should we limit ourselves? Really, it's an artificial distinction. And so what's going to be happening is that we expect that in 5 years we'll be in the clinic with at least one area that would not have happened but for USC's investment, and by the end of a decade we'll be in the clinic in 3 different areas, and we're going to have a whole infrastructure at the university to do this. And I should add that there's parallel efforts in viral-based and non-viral based gene therapies as well that will be coming out of USC. So it's a really exciting time to see a large institution that has this ambition, this vision, and resources, which is what many institutions are lacking, to be able to take a meaningful shot on goal.
Excellent. Well, we can't wait to see your progress. And if you missed Dr. Murray's presentation this morning, you can catch it on demand for 30 days on the ASGCT app. All right, see you at the next soundbite.